my great pleasure to present Kevin Madden, Senior VP of Platform Commercialization for Ginkgo Bioworks, who I'm sure is very attuned to explaining why he's doing these things. Kevin holds his PhD in biology from Yale, BA from the University of Pennsylvania, and he can explain what he did at Joule and other places. Please, Kevin.
Many thanks for the invite to say a little bit about Ginkgo Bioworks, and hopefully I can convey how these sorts of technology for writing DNA, amongst other tools, is helping to create value for a number of different industries, right? That's what Ginkgo is setting out to do. So I know Andrew gave a nice introduction into various elements of Ginkgo, and certainly spoke to the kind of aspiration and faith that existed, at least in the times that, in the early days that he was there. I know that I joined just over 4 years ago, so kind of at the tail end of that hope and aspiration stage when there were still 10 or 15 individuals, and a lot has happened since then, right? So at that time, since then, we've gone from 15 people to today we're just around 200 individuals, and going from bootstrapping to raising in excess of $450 million of equity, as well as, multiples on that in terms of expectations, right? So now the time is to deliver on the promises that we've conveyed, and we're doing so through a series of foundries that set out to make biology easier to engineer, really. So that's the fundamental principle of Ginkgo Bioworks. Is our mission is to make biology easier to engineer and to serve as a kind of a platform to enable new industries, as well as to improve existing industries, right? So to broaden the pie of things that are addressable through synthetic biology. We do so using a foundry-based approach, and what that really means is certainly a systematic and, where appropriate, automated way to do biology. We've now opened 3 different, what we call bioworks, so foundries for engineering biology, and in October we'll open a 4th. That'll be an exciting moment, because today we've entirely been focused on microbial systems, but with that foundry will enable our ability to engineer mammalian cells and other organisms as well. So exciting transitions, and certainly it's a platform that's built to broadly make biology easier to engineer, but you need to start someplace, right? And that was with microbes. And really the 2 assets, or at least technical aspects of the company, can be summarized in these 2 components. One is what we've called the foundries. It's really a physical asset, right? So it allows you to work on many projects in parallel and establish economies of scale that other industries, certainly more mature engineering disciplines, have done successfully over the years. And when you have that foundry and economies of scale, you also develop large amounts of knowledge, right? And so that comes in the form of genetic code base. And I like to think that, one of the wonderful things about nature is its diversity, but equally important is it's discrete, right? So there are only so many metabolic pathways that can be engineered to make different products. So the odds are, if you're working on 50 projects today, then the next one will be somewhat related to the next. Right? And that's the easiest way to improve probability of success, is to have done something quite like it in the past. So just a little bit of semi-promotional stuff, right? So we strongly feel that biology is one of the most sophisticated engineering tools on the planet, right? And a slide that's often used by our CEO is shown here, and probably looks a lot like the desks of many people here, perhaps, and maybe hopefully a little more organized than most, and I definitely have a cheaper watch, but if you look at that, it's the plant that's the most sophisticated object on that slide, right? It's self-repairing, it's self-assembling, replicating, renewable, resilient, right? And really it's also highly regulated, right? And so that's a large reason that biology is not so easy to engineer today, right? And that's something we all recognize, and we're hoping to develop general rules for engineering biology. But if we can do so, then we can enable entirely new industries and create a lot more value in the industries that exist today, right? And so that's really our fundamental approach. And we're applying it to a variety of industries, right? So with the objective to transform basic feedstocks, glucose shown there, and gas substrates at times into microbes, and enabled different industries. And know that Ginkgo has activities in all of those areas. But clearly, those are large existing industries today, but they're underserviced by fermentation and synthetic biology in general. And so that's really the crux of our objective, and to do so through different value chains and different partner networks. I think I'll dive right into some of our approach to organism engineering and really try to highlight examples of how DNA synthesis and associated tools are being used to enable different technologies. This is a slide that's probably familiar to many people that work in the general sector. So how projects are deployed in our foundries generally revolve around the design, build, test, ferment cycle, and really it's key to have as many of these approaches in one location, because really it's the power of the comprehensive approach rather than any of those in isolation, which isn't to mean that when technology becomes sufficiently mature, certainly we're not too proud to outsource those when it's either better done or more cheaply done through external means as well. But on the front end, and all this, a real large portion of why this has to be done in one site is because you need the learning and the software that enables that learning, such that you can leverage that into the next projects, right? But on the front end, we have large teams on really the experimental and DNA synthesis design side, right? Some of this can be done through rational means. So an example would be, you'll see examples that I'll show on rational design of enzymes, but many different approaches as well. And then once a large amount of DNA synthesis has been done, there's the need to assemble and move those products on through different microbes and on for testing. So kind of standard approach, just done at a much larger scale. And again, so much of this has been, and I think this audience is clearly very familiar with this, but since the turn of the century, the advancements that have been made in both DNA reading and writing, certainly reading was further ahead than writing for some period of time, but now the costs for DNA synthesis are much lower to enable different approaches, and clearly there are a further step change that we believe are possible. So just to kind of sum up the technical value proposition, of Ginkgo is really in two different forms. One is the physical scale of Ginkgo, such that one can undertake many projects in parallel, and in doing so you have the economies of scale, so your unit cost of doing a given event is much lower than if you're a series of projects, right? In other places that I've worked, when you'd have one project, perhaps it would be a team of 8 or 10 scientists, right? And for the next project, it'd be another 8 or 10 scientists. Really, the objective of Ginkgo is to uncouple the cost of development with the headcount required to do that, right? And in doing so, establishes economies of scale, and actually allows you to much more fully utilize the capital that you've deployed. Right, so you can afford to bring on more tools because you have a high capacity utilization, such that now you're getting a return on that investment. So that's kind of on the physical side, but certainly it's just as important to have advancements on the digital scale, and that's really all a summary of what I had referred to as the genetic code base. And this is just a couple slides that kind of reflects that economies of scale. And so on the y-axis is the number of foundry unit operations, and that could be as simple as a liquid transfer, it could be a PCR reaction, or some form of sequencing, NGS sequencing. And obviously on the x is time, and you can see a dramatic improvement on the throughput. We could also note that I said we raised $450 million over the last few years, so there definitely has been a significant headcount, right? So but importantly, it's not just spending more money. This slide is something that it's one of our kind of key performance indexes, right? It's what we call efficiency, and that's the number of unit operations performed per person in our foundries, right? And you can see that tracks very well with the number of unit operations. So we're really making good progress towards our objective of uncoupling how much we can do from how many of us are required to do that. Really, by doing this, we dramatically decrease the cost of development, or enable you to do more of that work and ensure the probability of success, right? So I'd be very honest and say today, the biggest challenge is that per unit capital invested, it tends to fail, right? And that's the biggest problem for attracting the next dollar to invest is they really don't have a line of sight to that return. But with Ginkgo's technology, we can essentially enable five times more shots on goal per unit capital, right? And by doing so, enable a much higher probability of success. So we can get to some examples that specifically talk about our ability to use synthetic DNA, right? And a lot of this is enabled by a variety of partnerships that Ginkgo has had, right? And DNA consumption at Ginkgo is on the order of 600 megabases per year or something. So a tremendous amount of DNA synthesis is done either in-house or through external partners, right? So those external partners include Twist Biosciences out in the Bay Area, a kind of real leader in the gene-length DNA synthesis category, and we continue to leverage that strategic partnership for that. And then a year ago, January, we acquired Gen9, and Gen9 is really differentiated in their ability to string together regionally long pieces of DNA, meaning kind of operon length or 10 kb or more. And by bringing them in-house, we enable them to fully integrate towards Ginkgo and our partners' objectives. So prior to the acquisition, they had 250 different clients that were all requesting things to be cloned into their unique vectors, right? And now all of that effort is tailored towards the needs of Ginkgo and our partners. So really it streamlines and allows us to optimize for things like turnaround time on DNA synthesis, which is clearly a big limitation today. So just some examples of how we use technology, and this kind of just reflects a general workflow. So as we work across the slide, on the left is just the ability to synthesize large amounts of DNA. It's not uncommon for a given project to consume on the order of 20, 30 megabases. That design can be informed by different things, right? It could be through protein structure models implied by the middle there, and certainly could be from just sequencing or synthesizing and or sequencing all the isozymes for a given enzyme that exists in nature, right? And then if you have a comprehensive approach that allows you to screen with, a readout that's facile enough to enable the testing of thousands of isozymes, and you have that full ability in-house, and that's how Ginkgo commonly does projects, right? Historically, it might have been that a given party would look at 10 to 50 isozymes of a given enzyme, right? And really just stay under the lamppost, right? But now with this DNA synthesis technology, you can look at a much broader set of diversity. So here are a few examples. This is an example where we're trying to use enzyme evolution to remove a key raw material in one of our partners' fermentations, right? Actually the most expensive component in that fermentation. So what was done there is we knew the enzyme class. It was widely present in nature, and that's basically a phylogenetic tree of most of the isozymes or candidate isozymes that had existed in nature, both in bacteria, fungi, and plants. And so what was done was we synthesized 900 or so different isozymes of this enzyme, cloned it into 2 genetic contexts, some different codon optimization, and then screened for activity. This screen for activity was done in vivo, but in many cases our screening we also have the capability to do most of this in vitro as well. And so this is one portrayal of the readout there, and clearly not all the enzymes have great activity, as there's a long lagging tail of things that didn't work. The arrow shows where their existing enzyme is, and you can see that we were able to discover many isozymes that had much improved activity and this enzyme has been put into an industrial host and is being used today commercially. So actually creating value out in the industry today for our partners. This is a similar sort of strategy for a different enzyme. So another output that shows nature's diversity. So it's an output that shows all the enzymes that have the ability or theoretical ability to have a specific biochemical mechanism, right? And a known example was on the left, and that's actually the seed that was used to design all of the DNA synthesis. And anything that has any color to it is an enzyme that had a hit in a primary screen, right? And so on the right there are known enzymes that have been well characterized and fortunately did show as strong positive controls in the screening. But I think the critical thing is that there are many isozymes that appeared even quite distantly related that one would not have expected, and some of those actually had improved activity over anything in the top row, right? So the ability to deploy large-scale gene synthesis, enabled the unexpected. So this sort of synthesis can be used in a variety of ways. One way that we often use it in this slide is quite dated, but it shows a number of different, industrial relevant enzymes. And on the light orange is shown at the time all the isozymes that were present in UniProbe, right? And note the x-axis is on the log scale, and then in the darker orange are all the assets that lived in Ginkgo's freezer, right? So for many isozymes, we have every enzyme that's possible, that's currently in UniProt or other sequence databases, and we continue to complement this with RNA-seq of our own to identify different diversity, right? And so these are assets that are in our freezer ready to be deployed against the next project, right? So there's no need to resynthesize, and it's just to reassemble perhaps if they're needed in a different microbe that has a different expression context. And it's not only to be used in kind of wholesale fermentations. We also deploy this sort of approach to improve industrial enzymes. And this is the same strategy in synthesizing and testing a wide array of different isozymes for a key industrial enzyme. And fortunately, there are facile readouts that both can be used to examine both catalytic activity as well as specificity for a variety of substrates, right? And so you can rapidly screen through a massive number of isozymes and even multiplex, looking at several different substrates and products in parallel. Next example is kind of an interesting example. It's one from a partnership that we have in the fragrance industry. So there are certain fragrances that leverage agricultural or natural products that are from substrates that are poorly available and often protected species, right? And so our partner wanted to develop a renewable approach and sustainable approach to making a particular fragrance. And this happens to be a terpene fragrance, and so we use DNA synthesis to both identify an isozyme that could kind of mimic the enzyme that was found in nature, and then we used a combination of rational enzyme design as well as exploring the limited diversity that exists in nature to both kind of tailor that fragrance. So a terpene synthase in this case makes a number of different products, right, including the one that's it's best known for. But through this design, we could steer that terpene synthase to make different portrayals of that scent, right? Because it had different distribution of different terpene products in that, right? And this is currently in a product that's being evaluated by our partner for further scaling. So just kind of a sampling of different ways that these sort of technologies can be used, from real basic commodity chemicals all the way to extreme high-end fragrances, right? So sometimes it's synthesizing the enzymes itself, and sometimes it's just a matter of looking at more context for the enzymes or sets of enzymes, right? And this is really just banging out large sets of operons or other genetic contexts and exploring diversity and having the readout there that can facilitate exploring that amount of DNA, right? So this was a commodity chemical that we were working with a large partner on, expressing 12 different enzymes, and clearly it wasn't optimal in the way that we first received the technology. So through a combination of varying the different isozymes, varying the promoters, varying the terminators, varying the kind of codon utilization, we're able to identify a vastly improved genetic context in a short period of time, right? And in doing so, this project is shown there relied on 20 plus megabases of DNA. So at Ginkgo, a variety of different industrial outputs. So probably the work that more often is promoted in corporate releases is our efforts on new product development. So to enable kind of top line growth for our partners, either in developing entirely new products or at least products that are new to those partners. Same set of tools are equally applicable to kind of strain improvement. Though certain things there, like using DNA sequencing to better understand the past changes that have been made, just both good and bad, and then often going back with more advanced technologies to only introduce the good changes into the production organism as part of the strain improvement approach. The same approach that's used to make new products by constructing complex multigenic pathways is equally applicable to expression of enzymes as the actual product. And then sometimes it just makes sense for partners to do their discovery or basic R&D at Ginkgo because we have the economies of scale, right? So instead of booting up an entirely new R&D project, they can invest their capital in a more mature R&D context and both get results faster and less expensively. So I've kind of stated it a number of times, but Ginkgo's business model is to be a kind of enabling platform to different industries, and thus all of our value is created through our partners, right? And so we have to develop partnerships that share the aspirations, or else the pie is not going to grow that much, right? And so shown on this slide is a set of our publicly announced partnerships. Today we have 20-something different partnerships, but don't have the ability to disclose all of those, and working on upwards to 50 different products. So that's kind of like a sampling of the scales that we're able to achieve through the technologies and the platform developed to date, and we look forward to launching more bioworks to enable more technologies through partners. And clearly, all of the partnerships don't have to be in the form of industrial partners, right? So there are a number of different, so we also have technology partners, right? So Ginkgo has announced strategic relationships with companies like Genomatica, who provides kind of a complementary approach to technology and kind of more rigor on the back end of the development process, right? Similarly, we have strong relationships with academic and government institutions, right? And I think that's part of the reason that attracts me to come here is to hear a different perspective and to see how Ginkgo may assist or help inform decisions throughout a variety of sectors, right? So thank you very much for your time and attention.
So Kevin, thanks a lot for a very different perspective on the topic. Questions? Please, yeah.
Yeah, we'll see if I can morph this into a question for you and not just a heavily weighted statement. But from, a prokaryotic perspective, most of the examples that you gave of gene synthesis were synthesizing single genes or metabolic pathways in order to produce novel functions, either catalytic or otherwise, in a host. Do you think that in the context of GP-Write, it'd be worthwhile right now to have a component that is synthesizing entire genomes versus synthesizing smaller fragments and doing more functional analysis of the genomes themselves before we actually move into an entire genome? And do you think that those projects where you're looking at portions of a genome should be able to fall under a GP-write mandate if we're going to move into entire genomes later on?
Yeah, no, it's a great question. Definitely one that I recognize sitting in the audience here is that our synthesis does tend to be, a given enzyme we're stringing together an operon, right? 20 kb is starting to kind of push the limits of our standard assembly, something that can live in a plasmid but clearly we also have projects in Saccharomyces and other organisms where you can string together much larger pieces through combination, but not on the order of a chromosomal scare. That's just not the sort of projects that we've taken on, right? So I definitely welcome comments and ideas on how kind of the power of our synthesis and perhaps some of the assembly approaches could help enable some longer assemblies, but it's not a real focus of Ginkgo today.
Speckler?
Thank you.
Maybe you can stand up and ask the question.
So you did mention that one of Ginkgo's capabilities right now is sort of the ability to use data that you've collected to sort of predict what the phenotypic expression might be just given like a particular DNA sequence.
Mm-hmm.
Is this a data set that's sort of weighted towards prokaryotic organisms? Does it work well on different organisms from, say, more complex genomes? Does it have? Can you predict how genomes might turn out, or how genes might turn out in sequence? Not really sure how to spell this one.
Yeah, no, it's a great question. And certainly our data sets contain large amounts of plant and fungal and even mammalian data as well. So but I would say we're a long way from being able to accurately predict the function, and really that's part of Ginkgo's core existence, is believing that we need empirical data to inform future, predictive function, right? And so we're really focused on tools to enable generation of a lot of empirical data, even if it is not directly relevant. I hope some of that data still correlates with what would be observed in a different context. So I guess I'm waffling back and forth, but know that like my personal opinion is that we need to generate much more data before we really can, have a strong predictive function, and that's what Ginkgo's focused on doing.
All right, thank you.
Yep.
Please, over here.
You mentioned that you come across examples of unexpected effects. I'm just wondering, do you have specific strategies for combinatorial biosynthesis? So, if you have an enzyme that you find an isoform that is more optimal, you combine it with another enzyme in a different part in the pathway, do those generally tend to work additively, or do you find conflicting effects at different steps?
Yeah. And I'm probably not the best to answer this, but a couple points, right? And the trees that we've shown, our basic approach is we definitely look under the lamppost as well, and then we try to pick a certain number from each node to ensure that we source sufficient diversity to find the unexpected within the balance of how much a readout can look at. Clearly our design does tend to skew when there's co-expressed to have either common organism. So like we'll bias our synthesis to go towards things that one would just on first principles think play better together, right? But we don't exclude. So just like we do in sourcing the primary diversity, we still paint in some of the unexpected combinations to see if something unexpected is there. So
I had to step out for a minute. You talked about it already, but so a lot of what we talked about is enzymes and pathways, but the jargon people use is the chassis of the host.
Mm-hmm.
So do you just take those off the shelf, or do you want host engineering? I think a lot of what the GP-Write program can and should be looking at is this genotype to phenotype relationship.
Mm-hmm.
And in industrial biotechnology, you need to have a certain understanding of that. So do you guys do this at all, or?
Sorry, I'm not.
So for example, do you ever? So you need a host that's tolerant to low levels or high levels of acid.
Mm-hmm.
How do you deal with that? Do you engineer that? Do you just find an organism?
Yeah, no, it's a great question. And so can kind of go at it from a couple ways. So you talk about chassis organisms, and chassis organisms can be, you know, built up to push flux in a given metabolic direction, right? And certainly we have those, and then, but chassis organism isn't so plug and play. You have to make sure it's all balanced just right to enable the rate limiting portion of that, right? And then so that would be one type of chassis. Another type of chassis that's less currently based on first principles would be one that's has a physiological desirable trait, pH tolerance, or, you know, alcohol tolerance, or whatever it is, right? And so it's pretty hard to teach a workhorse organism to have acid tolerance, right? That's a very highly multigenic trait, right? So you have to develop our current approach is to take on a large number of organisms, right? So today we have more than 15 different species of organisms deployed across you know, different industrial yeast, fungi, gram-negative, positive sporogenic bacteria as well. So really, we believe that in order to, at the end of the day, hopefully we can have chassis workhorse organisms for these complex traits, right? We're not there today, so we go to organisms that inherently have a favorable disposition for those traits. So.
Let me just ask you a little question myself. So, having run industrial partnerships with universities, it was always an issue for us, the stuff that you had to keep inside the lab for intellectual property, competitiveness reasons. Do you have a sense of when you're dealing with academic partners, if you like, how many years horizon you like to be out, or sort of how far off of your core strategic business you like to be? I sort of draw it as a bit of a bow wave, but past a certain point, you're really working with the academics. Because what you're talking about is right now, and I think what we were talking about with Bogomil was like 5 to 10 years out, and I was just wondering where your sort of sweet spot for academic collaboration is.
Yeah, so I would think that it's more at the, so Ginkgo's intent is not to be at the leading edge of technology development. We want to be the implementer of the best in class, right? And so to the extent that we can encourage exploration and innovation, right, and kind of be a beta tester of that, and with some form of agreement around it. It doesn't have to be exclusive, right? So that's the sort of area that I think is best suited for our interaction with academics. So it gets more complicated in other areas because all of our value is created through our partners. So now we have 3, and that's a tougher dance, right? And so I think relationships more on innovation and that sort of area are better suited.
Excellent. Yeah. Kevin, thank you so much.
No, thank you.